So last topic for today, I'm going to go over gel electrophoresis. So gel electrophoresis, it's a method used to separate different compounds based on their size and charge. So this is most commonly used to separate DNA, RNA, and different proteins. So how it works is you place the mixture of either like DNA, RNA, or proteins on the gel, and then you apply an electric field across the gel that has a negatively charged side in the cathode and a positively charged side known as the anode. So because of the electric field and the two different charged sides, the molecule will travel through the gel to either the cathode or the anode based on their charge, but they will travel at different speeds depending on physical properties like their size and their charge. So the larger molecules will move slower than the smaller molecules just because of the size, and then molecules of the same size that have greater charges will travel faster than molecules that are the same size but have a smaller charge. So that's just the general basis of gel electrophoresis. So gel electrophoresis usually, most likely they'll use polyacrylamide gel. So polyacrylamide gel electrophoresis, most commonly known as Page, is like the basic method for gel electrophoresis. So like I said before, you separate the compounds based on their size and or charge using an electrolytic cell. Because there's a cathode that is negatively charged and an anode that is positively charged, the cathode will attract the positively charged substances while the anode attracts the negatively charged substances. So the compounds will travel to the side with the opposite charge, and the gel separates the compounds based on their size and charge. So native Page is the most basic form of Page. This is used to analyze a protein in its natural state, so nothing is done to the protein or the DNA or RNA. The protein will remain in its native shape and maintain its secondary, tertiary, and quaternary structures, and the DNA, RNA will keep their original shape and structure as well. This separates the substances based on their size and their charge. So as you see at the bottom, that's the general setup for gel electrophoresis. You have the power source that acts as the electrolytic cell. You'll have the gel in the structure that separates the sample, and then there's wells in the gel that you deposit the sample in. And once, as you can see, the power source, it's connected to the two different sides of the gel, and once that power source is turned on, electric field will be made through the gel, and it will force the samples that were placed in the wells to travel across the gel. So that's native Page. SDS Page, this is when you use a detergent called sodium dodecyl sulfate, most commonly referred to as SDS. So this detergent is used to denature the protein and adds a number of negative charges that are proportional to the size of the protein. And this is done to create an equal charge distribution. So none of the proteins will have greater or a lesser charge. They'll have the same charge, same uniform negative charge across the protein. This is done in order to give them a uniform charge. So you separate the proteins based on their size alone. So native Page, it's it separates the proteins based on charge and size. But once you use a detergent that creates a uniform charge, it will only separate the proteins based on their size. So SDS Page is when you want to just separate them based on their size alone. So SDS Page, as you can see, there you have the 3 different proteins on the left, A, B, C, and A, B, and C. And after you use SDS on it, all the proteins will have a uniform negative charge across the protein. And when you run a gel electrophoresis with it, the proteins will travel across the gel, and because C is the smallest protein, it will travel farther in the gel than A or B, because A or B are a lot bigger than C. So this separates the molecules based on their size alone, and the smaller molecules will travel farther in the gel than the larger molecules. So that's SDS Page. So there's a form of SDS Page called ReducingSDS Page. So besides adding SDS that completely denatures the protein, except at places where there are disulfide bonds, you add a reducing agent that reduces the disulfide bonds as well. So the reducing agent will break the bonds in the disulfide bond, and this causes all the higher structures of a protein to be eliminated. So there are no tertiary, quaternary, or secondary structures anymore. It's just the basic primary structure of the proteins. So reducing SDS Page, you add SDS and then a reducing agent to have the protein in just its primary structure.